Our method uses whole strain selection and fluorescent markers for the efficient and seamless generation of recombinant Vaccinia virus in a variety of cell types.
This technique combines the speed of homologous recombination with the scarless nature of transient dominant selection. In addition, we use host range selection to eliminate the possibility of chemically induced changes.
This method can also be used to modify Vaccinia virus or other poxviruses to express foreign genes, for example, to generate vaccines.
If you're performing this protocol with fluorescent selection only, you must ensure that you're screening enough viruses to detect the relatively rare recombinants without PKR selective pressure.
This method relies on the gain and loss of fluorescent markers to successfully select viruses that have acquired the recombination cassette and to detect viruses that have scarlessly recombined.
To generate a recombination vector, first add 17 microliters of DNase-free water, 1.2 microliters of each primer, 5 microliters of 5x PCR reaction buffer, template DNA, and 0.5 microliters of DNA polymerase to 1 PCR tube per amplicon. Add additional DNase-free water to bring the final volume in each tube to 50 microliters, and place the tubes into a thermocycler. Melt the DNA at 98 degrees Celsius for 30 seconds before using 25 rounds of a 3-step PCR protocol as indicated. To visualize the amplification products on a 1% agarose gel, add 10 microliters of each DNA product and 2 microliters of loading buffer to each well. Run the gel for 1 hour.